Hello, welcome to single-cell multiome analysis series with SciDAP. In the previous video, we started the single-cell ATAC clustering analysis, and in this video, we will look at the output. As previously for RNA, we could see the distribution of cell numbers between clusters and see the distribution of QC metrics. Interestingly, we have one of the clusters that has very broad number of peaks and ATAC fragments in peaks. You probably might remember this sample from the filtered analysis. Its peak was shifted. For resolution 0.5, we also could see interesting things are going on with the cluster 7. We should keep in mind that the resolution 0.3 and 0.5 have different number of clusters. Probably this cluster had very low number of cells. Could we trust this resolution? Probably not. So between these and these resolutions, there is only one cluster that is additionally produced. However, let's look at the clusters themselves. It doesn't look suspicious at first, but when you look at the silhouette scores, the average silhouette score for all the clusters is actually negative. It means that the algorithm is not sure to what cluster cells must belong to. It is only certain about the cluster 2, which has very nice and high silhouette score, and other clusters are all on the negative side. So cells could be equally likely to be in each of these clusters. And the same for other resolutions. It means that either there is not much diversity between the cells' DNA accessibility, or the clustering algorithm couldn't assign cells to any of the clusters because of the parameters that we set. There are more tabs that we were reviewing in the single-cell RNA clustering. There is split by dataset, split by group. There are also clusters that are differential between our group, recirculating cells and the TRM cells. Here is the cluster composition. And this is the gene of interest coverage. It will show you the genes that you provided here as transcripts and the peaks. And each track represents the cell cluster. And here, because we provided RNA expression as one of our previous inputs, we could even see the expression of this gene and how it correlates with accessibility. And it might be different for every resolution you provided. Then there is a peak markers table, just like we saw it for the single-cell RNA-Seq. And let's return to our silhouette scores. I would like to try a different integration algorithm to see if that will affect the results. So to do that, we need to go back and restart our single-cell ATAC-Seq dimensionality reduction analysis. So I already restarted it with Harmony. I provided it as an integration method. And additionally, I changed the parameter, which called the minimum percentile of highly variable peaks. I set it to 10, meaning that 90% of the highly variable peaks will be considered for integration, scaling, and dimensionality reduction. Additionally, I provided the custom patient as the batch effect parameter for Harmony. Finally, normalization method. I changed it to log-TF, log-IDF. Information about this method you could read in the papers that mentioned here. So this is my second run. Let's look at it. Looks different from what we saw initially. Silhouette scores look better now, even though most of the clusters are still below this red dotted line. But this time, at least 3 or 4 clusters are defined more confidently. So probably the ATAC-Seq profiles are very similar to each other. And Even though it's not perfect, this dataset is a good demonstration of how using different parameters you can change the cluster analysis. There is information that we already looked at, the peak markers, and also there is UCSC browser where you could see better those peak markers that I was talking about. You could sort them based on the p-value or log-fold change. Sometimes it is hard to say using the coordinates what gene is nearby, but there is a solution. You could switch to the Feature tab and search for any gene of interest, and it automatically shows you the list of nearby peaks downstream or upstream of the promoter. You could even highlight multiple or make it split screen. Because we use the RNA clustering as a previous sample, we could also see clustering for RNA in this UMAP as well. So this was the quick review, and in the next video we will start the step that called WNN clustering. In that step we will combine signals from both ATAC and RNA together using weighted nearest neighbor method. Thanks for watching.